Hello, everyone. So today we're going to talk about molecules. In Chapter 2, we discussed atoms, bonds, atoms making bonds, and when they do that, they make molecules. So this is the next step, so a larger step that we're taking, and these molecules will help us do all kinds of cell functions. A cell is mostly water. The result of the cell consists mostly of carbon-based molecules. And generally, you're going to have a carbon backbone with carbons connected together, different molecules coming off of them, and this is organic chemistry. That study is called organic chemistry. We call it organic because it's carbon-based, and everything with carbon would be organic chemistry except monoxide, so CO, and dioxide, CO2. Those are abiotic, those aren't living factors. So moving on to carbon chemistry. Carbon can make 4 bonds. This is important because it can make really complex molecules, which is important for life. It makes covalent bonds with hydrogen, and then electrons are shared between carbon and hydrogen. We got into this a little bit last chapter. Different examples of carbon backbones. Carbons attached with lots of hydrogens are right here. And then we have double bonds, which change the structure slightly down here. And then we have ring-based bonds. And these are examples of what steroids would look like. So molecules with C's and H's, so carbons and hydrogens, are hydrocarbons. An example is methane. We have 1 carbon and 4 hydrogens. And we would write this as CH4. And this makes good gasolines. The carbon bonds are full of energy to help power things up. And then depending on what atoms we have to put together, they're going to give the molecules different shapes, which give it different functions. In anatomy and physiology, the anatomy or the structure determines the physiology. So what it looks like will determine how it functions. For example, neurons in this one, neurons are sending neurotransmitters which match up with these little receptors. So the neurons, receptors, or neurotransmitters and receptors. They fit in like little puzzle pieces. What's happening here, for example, our hormones are making changes, and the cells where changes are made have receptors that fit the hormones so that those changes can take effect, etc., etc. The unique properties of organic compounds depend on not only its carbon skeleton, but also on the atoms attached to the skeleton. So what we have here is we have C's and H's that we've already gotten used to, carbons and hydrogens, and then we have other groups which are called R groups or functional groups, for example. In hydroxyl OH, you have an alcohol right here. In carbonyl groups, our carbon double bonds with an oxygen. Here it's double bonded a little bit differently. Then we have amino groups, which have this nitrogen right here. And then we have carboxyl groups, which are also called carboxylic acids. And we have double bond on the oxygen here. Oh, I lost my mouse. Here we go. The oxygen here, and has a little alcohol at the end. And all of these different ones change the function and shape of carbon-based molecules. So moving on to giant molecules from smaller building blocks. So generally we don't see any molecules one by one, but rather in large scale. So macromolecules, and macromolecules are molecules that we can actually see with our eyes. Examples are proteins, DNA, RNA, which are nucleic acids, proteins, we have lipids, which are fats, and then we have carbohydrates or sugars. So the way we make macromolecules is by putting a bunch of them together. So remember, they're polymers. Poly means many, and polymers are made with a bunch of monomers. So mono means one molecule. Many molecules are made with one molecule. What we do is we keep adding monomers together, which make a large chain. It's like if you're stringing Fruit Loops. Each Fruit Loop is a monomer, and by the time we get to a finished Fruit Loop string or necklace or bracelet, whatever, we have a polymer. So the finished product is a polymer, and then each of those little Fruit Loops is a monomer. And the way we link these is with a process called dehydration synthesis. And the way this works is dehydration. Everybody knows what that means. It's when you need water. So we're dehydrating this molecule. We're taking the water out of it. So we take out a water, and then we are able to build it. Dehydration, remove water. Synthesis means build. All right. For example, glucose is a monomer, and it forms a polymer, etc., with dehydration synthesis. So to put it back together, so the opposite of dehydration synthesis is called hydrolysis. That means we need water back in to break down that molecule. So we build it, dehydration synthesis, opposite of that, hydrolysis. So break that baby back up. Hydro means water, lysis, breakdown. So next, we're going to move on to the 4 biological molecules that are essential for life. So we have carbohydrates, which are sugars. We have lipids, which are fats, and we have proteins and nucleic acids, which are DNA and RNA. Let's start out with carbs, carbohydrates. Everybody hears about these. Carbs are small sugars. They're monomers, and we have these like in soft drinks, candy, anything like that with small sugar molecules. And then we have large, long starch molecules, which are in pastas and potatoes, etc., etc. And these are just different kinds of carbohydrates right here. We have monosaccharides. Mono means one. Remember, saccharide, sugar. So monosaccharides are simple sugars. So think of one as being a simple number, I guess. That would be a good way to remember it. To remember that monosaccharides are simple sugars. We have glucose, which is found in sports drinks, and we have fructose, which is found in fruit. So fructose found in fruit. They usually have, whenever we see glucose and fructose written out, it's usually a formula like this. This means they have 1 carbon, 2 hydrogens, and 1 oxygen. And the reason they are in parentheses like this with an X out here is that this is just 1 molecule, and if we have many of them together, we just put an X down here and write down how many we have together. So if we have a 5 carbon sugar, let's say, we would write a 5 down here, and we'd multiply every little number down here by 5. So like, let's just pretend there's a 1 right here, 2 right here, 1 right here. 5 carbons, it'd be C5H10O5. If we had 6 carbons, so remember glucose has 6 carbons, it would be C6H12O6, which is what you'll hear a lot of scientists call glucose. We won't call it glucose. They'll just say C6H12O6. Here we have glucose and fructose. They're called isomers, basically because of this. They each contain the same atoms, but the atoms are arranged differently. So you'll see the double bond with the carbon is up here. The double bond with the carbon is in a different place right here. But they have the same amount of hydrogens, oxygens, and carbons, just placement of that carbon. Like, for example, the first carbon has a double bond in glucose, the second carbon has a double bond in fructose. So again, isomers have the same formula, but the atoms are in a different arrangement. When those get into the water, so aqueous solution or aqueous solution, remember aqua, water, we get a lovely ring structure. And we usually don't show all of these details. We'll abbreviate it like this. However, for the purpose of this introductory class, we will see it like this, just to have it broken down for us. Our cells need carbohydrates, and they're constantly moving through our blood. They're being monitored by our pancreas to make sure we have good blood sugar levels so that our cells can get glucose to make energy. So disaccharides, we have two monomers put together. So a disaccharide comes from two monosaccharides. So two saccharides from one saccharides, basically. Two sugars from two simple sugars. Glucose plus glucose in this example makes maltose. And remember what this process is called. We take the hydrogen out. Or not hydrogen, we take the H2O, we take the water out, so it's dehydration synthesis. So take out a water to build a sugar. Another example would be, well, let's go back up here. Another example would be lactose, which is sugar in milk. People who are lactose intolerant, they can't break down the bond between those two sugars, which needs to happen to get them to the rest of our body. But in lactose intolerant people, that doesn't break down and goes through intestines, and the bacteria in our intestines just love it. And they're the ones that use it all up, and they're the ones that break it down. And that's why lactose intolerant people have just super uncomfortable gases and, other effects thereof. Oh, I clicked a little too far. Okay, here we go. The most common disaccharide that we come into contact with is sucrose, and that's just table sugar, and table sugar is in so many different things. Here's an example of glucose and fructose, and they make sucrose with that little binding right here. And the way this is made, this little diagram right here shows you how the sugar cane goes through this structure and gets broken down into these disaccharides. Basically what happens is they get squished and boiled, and we get these disaccharides in the form of table sugar. Crazily enough, we eat about 45 kilograms, which is almost 100 pounds of sugar a year. So crazy. That's so much sugar. 100 pounds of sugar. All right, let's move on to polysaccharides. So polysaccharides are mini saccharides. Remember, poly means many. So we have mono, which is 1. We have di, which is 2. And then we have poly, which is many. And these are just super long chains of sugar units. And again, these are polymers of monosaccharides. So this is just a bunch of monosaccharides put together. And we call these complex carbohydrates because it is a lot of chains. We'll see this a lot in starches. Bind them together with dehydration synthesis, get long chains. We have different examples of polysaccharides. So I just mentioned starches. We have starches in the form of potatoes and different pasta. And here's just a glucose monomer, little example of that chain. And we have glycogen as a polysaccharide, which is an animal muscle tissue. And we have cellulose, which is inside of plant cell walls. It's usually in woody plants, and we generally can't digest these. So what does that sound like? That sounds like fiber, right? And it's good for us to take in fiber. So in plant cells, we have rigid walls, and plants use these rigid walls to store energy. In people, we have the liver, right? And the liver stores glycogen. So if our blood sugar ever gets low, our hormones will tell our liver to release more glucose and get our blood sugar back to a normal level. So cellulose is dietary fiber, like I just said, and we have a lot of it in this world. We can't break down cellulose, but different animals can because of the bacteria that's in their digestive tract. For example, cows, and that has to deal with hind guts, etc., etc. Alright, so one of the most important things about sugars are their interactions with water. Sugars are hydrophilic, remember? Hydrophilic means, or I don't know, did we talk about, we did talk about hydrophilic and hydrophobic. So hydrophilic is water loving. Hydrophobic would be water fearing, so it would try and stay away from water, kind of like oil does. But sugars are hydrophilic, which is water loving, which means they dissolve. They're soluble in water. So I just said hydrophobic were different oils. Well, that's a good segue into lipids. Lipids are fats and oils, and they are hydrophobic. They do not mix with water. They don't like water. They're terrified of water. Here's a great example. So these are just different examples molecularly, and how this waxy cuticle makes this water droplet stay on the leaf instead of getting absorbed by the leaf. And then here, oil and water, which is what we're used to. So if you want to do this experiment yourself, get a glass of water, drop some oil into it, and watch it not mix. But some examples here are also fats and steroids. So we have these fatty acid tails that latch onto a glycerol. So when we eat food, if we don't burn these calories, they can turn into triglycerides. So triglyceride right here. And one measure of health is your triglyceride level in your blood. So if any of you have had blood tests, they look at different HDL and LDL levels in your triglycerides, but we don't have to get into details about all of that. Alrighty, so that being said, we do need fats. So fats are where we have our energy storage, where we can make energy, exactly what it sounds like, and store it, for future use. It gives us cushioning, which helps protect areas around our organs, and it provides us with insulation. For example, someone who's really, really, really thin has problems maintaining normal body temperatures. It can go from really hot to really cold really fast. Also, when we talk about fats, we look at different types of fats, like saturated versus unsaturated. We've all heard these, we've read them on food labels, but what do they mean? So let's delve into some details to get a little deeper understanding of what we're reading about on all of our food labels. So in this chain, this saturated fatty acid chain, you see all of these carbons that are saturated with all of these hydrogens. You don't see any double bonds, you just see all of these single bonds around. In unsaturated, we have this double bond, and which makes the carbons have less hydrogen on them. Notice that it's double bonded here, and these two do not have a hydrogen attached. Well, they have one, but not another one. This just little bond right here creates this bend and makes it more complex. It doesn't allow everything to sit on top of each other as well as it does in the saturated fatty acid chain, right? So they don't, it's not as easy as it is in the saturated fatty acid chain. So let's talk about different types of saturated fatty acids. They're flat, straight carbon chains, and they have tons of hydrogens that are stacked together nicely like we just saw. And they will be solid at room temperature because they fit together so well. And different examples we have here, our bacon, eggs, and just think about all that greasy deliciousness, right? Whereas unsaturated fatty acids, they don't sit together as nicely. They have those double bonds, they have that bend, etc., which it doesn't let them stack on top of each other as easily. So they're a little bit more, complex, and they are oil at room temperature. So with saturated fatty acids, we have animal fats, so things like butter, and then usually plant oils will be low in saturated fat, so corn oil, olive oil, different things like that. So let's move on to steroids. They're another type of fat, another type of lipid, and they're different. They are not long chains, they're actually these ring structures. And they're all derivatives of cholesterol. Cholesterol is the base, and we change the structure to make different steroids. So, for example, we have sex steroids like testosterone, estrogen, progesterone, stress hormones like corticosteroids, and they're all important in sending signals to different cells. And next we have proteins. Proteins are super variable in structure and function, and they're made out of amino acids. So a protein is a polymer. It's a long molecule, and it's made out of building blocks called amino acids. Different amino acids cause proteins to have different shapes and functions. And we have 4 types of proteins. We have structural proteins like keratin in hair. We have storage proteins for energy. We have contractile proteins, which are like in muscles. And then we have transport proteins like hemoglobin in the blood. Hemoglobin is a protein that helps us transport oxygen. And all of our proteins are made from a common set of 20 amino acids. Some are small, nucleophilic, hydrophobic, so water-fearing, aromatic, acidic, sorry about that. Whoa. And so forth. So 20 different amino acids. And the way they're put together will determine a protein structure and function, so what it looks like and what it does. Each amino acid will have a carbon atom with an amine group, so remember, or you can call it amino or amine, both are correct, and it'll have a carboxylic group, or carboxyl group. And then this side group right here is also called an R group. I think earlier we may have called it a functional group. It's all the same thing. Side group or R group. The side group determines what amino acid it actually is and what it looks like. And it determines if they're hydrophobic or hydrophilic, for example, in these two. Or it could determine if it's any of these examples. All right, proteins as polymers. So just like in the others, you take monomers and you put them together with dehydration synthesis. So take oxygen out to put these different amino acids together. In proteins, this bond has a different name. We don't, just call it a chain. We call it a peptide bond. So if you see or hear peptide bond, you will absolutely know that we're talking about a protein. Let's see. A small protein, it can be referred to as a polypeptide, which just means it has a bunch of peptide bonds. So you'll hear polypeptides a lot, and you'll again know that we're talking about a protein. And our body has tons of different proteins and protein structures. So, for example, this is a primary protein structure, and this is just a chain of amino acids. A primary structure is one-dimensional, so the order of amino acids is put together specifically. So there is a reason for that specific sequence of amino acids. So each of these is a different type of amino acid. If you change one amino acid, well, not you specifically, but if one amino acid is wrong in a chain, you can get different diseases. So, for example, here we have a normal can't talk today. For example, here we have a normal red blood cell with this protein chain. Normal hemoglobin, normal red blood cell. Down here we have a sickled red blood cell, and this is what that chain looks like in a sickle cell hemoglobin. So that's what the amino acid chain will look like. The amino acid is different right there, and just that one little change makes your normal red blood cell go from this to this, and this is called sickle cell anemia. There's one mutation point, so a single mutation point, and it changes what hemoglobin looks like and gives it this. The reason we call it sickle cell is this moon shape is referred to as a sickle shape. And later, we'll talk a lot about this disease in countries affected with malaria and different adaptations therein, but that'll be later in the semester. Some pretty cool interactions happen between this disease and the countries. But basically, sickle cell red blood cells are smaller than normal red blood cells, so you can see, and they don't carry oxygen as well. By the way, a good way to remember differences between proteins, sugars, fats, lipids, and nucleic acids is proteins are going to end in IN, so hemoglobin, protein. Sugars usually end in OSE, so fructose, glucose, and then fats and lipids will end in ONE. So different hormones is an easy way to remember that. So let's just get back to protein structure. We just talked about primary structures, so one-dimensional chains of amino acids. Then we have secondary structures, which are two-dimensional with alpha helices and beta pleated sheets. Then we have a tertiary structure, which are three-dimensional, so see 1, 2, 3. And tertiary structures are going to be polypeptides, just a single polypeptide. And then we have quaternary structures. So this is the fourth type, and it's not 4D, it's still 3D, but it's quaternary structures are just a bunch of tertiary structures put together. So here we have that one, primary, 1. We have 2, which is secondary, with this alpha helix and beta pleated sheets. That's what those look like. Then we have C right here, which is tertiary, number 3. And then we have this quaternary structure, which is our fourth structure, and it's just a bunch of tertiaries put together. Another example of one with hemoglobin. So we have primary, again, list of amino acids. Secondary parts will be alpha helices, other parts will be beta pleated sheets. We have tertiary, that has one subunit of hemoglobin. And then for hemoglobin to take on its final structure, we have this quaternary structure, which is a bunch of tertiaries put together. I will say some proteins will stay in the primary structure, some will stay in secondary structure, but yeah, this is just one example. So what determines protein structure? A protein is sensitive. It gets its feelings hurt very easily. But no, it is sensitive to its surroundings and due to different hydrophilic and hydrophobic properties and all that good stuff. If a protein is put in unfavorable temperature or pH, that protein will unravel and it will lose its shape, and that's called denaturation. For example, if you're cooking an egg, that egg white, once it turns white, it's denatured and you can't get it back into its original form, right? So think about frying an egg. Once that egg is fried, there's no going back. It's denatured. It's over. All right, moving on to nucleic acids. So this is our last type of biological or organic molecule that we're going to talk about today. And this is how we store information and send it to our other cells. So we have DNA right here on the right. It's double-stranded, and it passes on information in the nucleus from cell to cell. And then we have RNA, which is single-stranded, and RNA will code for our amino acid order. So nucleic acids are polymers of nucleotides. We have a phosphate backbone in both. We have sugar and we have nitrogenous bases. So let's look at that here. So we have this phosphate backbone, we have this sugar, and then we have the nitrogenous base right here. And the nitrogenous base are the encoding pieces. So here we see A, G, C, or T, adenine, guanine, cytosine, thymine, and they make up the ladder steps. So what am I talking about? These are the ladder steps, or rungs of the ladder. So each DNA nucleotide has one of the following bases: adenine, guanine, thymine, and cytosine. Adenine and thymine will always undergo complementary base pairing, so A and T will always go together. And then cytosine and guanine will do the same. So one purine will always bond with one pyrimidine. Oh, adenine and guanine are our purines, and cytosine and thymine are our pyrimidines. So A and T will be together, G and C will be together. And nucleotide monomers are linked in this long chain, which you could refer to as polynucleotides, but we're just going to call them DNA strands. And then, again, we have this sugar phosphate backbone that joins them together. Then two strands of DNA will come together to form this double helix. And like I said earlier, A will always bind with T and C will always bind with G. And that's called complementary base pairing. So moving on to RNA. It's a little bit different from DNA. Its sugar has an extra OH group. And then instead of thymine, like we saw right here, the T, we actually have uracil, so U. and then your cell will still be a pyrimidine. So let's talk a little bit about DNA discovery. We can use these DNA to look at how closely organisms are related. For example, through DNA discovery and looking at different, DNA in different animals, we discovered that we are more closely related to chimpanzees than eastern bluebirds are to blue jays. Isn't that crazy? So learning about DNA has just expanded our knowledge of these different relationships. Then we can make and with these different relationships, we can make a phylogenetic tree and see where we've branched off. So remember from one of our first chapters, this is the point really long time ago where our common ancestor is, and then just branch off everywhere. It looks like the frog hasn't had much variation, right? Mouse branched off around 80 million years ago, and then we have this last common ancestor about 26 million years ago before we branched off into different types. Alright, and that is it for our chapter on molecules.